To you my NYU Tandon undergraduate summer research program project that I've been doing over the past summer with Professor Nikita Grigoriev. So a little bit of background information. My research involves the identification of specific types of bacteria and prokaryotes in bodies of water that are used for things like swimming, fishing, and surfing that might have been contaminated from sewage or wastewater. And this is important because a lot of the times when the water is contaminated, this can lead to the presence of E. coli and Shigella, which are types of bacteria that can cause serious waterborne illnesses that affect your central nervous system, your skin, respiratory system, and etc. So essentially what my project involves is 16S metagenomic sequencing, which is looking at the 16S ribosomal RNA, which is a structure that is found only in prokaryotic organisms, such as bacteria, but not in us since we are eukaryotic organisms. And it looks at 9 regions. Since this structure is actually highly conserved, it actually has 9 regions that differ based on the type of organism that you are looking at. And from there, using specifically the V3, V4, and V7 variable regions, we're going to try to identify the specific type of bacteria that is present. And what we do is, after the samples have been collected and filtered, we're going to try to isolate the DNA from the water. After isolation of DNA of the water, we are going to try to make sure that there's enough copies of the DNA, or that there's enough amount of DNA that is present in our samples. And we use this through a PCR reaction, or polymerase chain reaction, which basically acts like a printer and makes a bunch of copies of the DNA. And from there, we're going to run a gel. The reason why we run the gel is to make sure that the DNA is in the proper region, it's in the proper amount of length, and that we did isolate our DNA correctly. So we match it up. We match up the bands, or essentially the lines that you're going to see on the gel, with the DNA ladder, which allows us to see how much base pairs and to make sure that everything has gone smoothly. After that, what we essentially do is we're going to prepare the DNA and start preparing the library, which is actually just what it sounds like. Just like a library contains or stores books, this sort of library the DNA library is just going to store all of our DNA that we isolated and filtered. And what it does is it's essentially going to start to index or categorize the DNA so that we can sequence the DNA. Sequencing the DNA is going to allow us to compare it with other known genomes or other DNA libraries of bacteria that we already know. Using those sequences, we compare with our sequences that we got from our samples and are able to identify the specific type of bacteria or organism that might be present in the water.